Welcome, everybody. This is Julian from Straight from a Scientist, and I'm here with a very special guest, Irene Gonzalez, one of my classmates in my master currently. So, Irene, you want to introduce yourself?
Hello. You can also call me Irene.
Irene, yeah, we make fun of her because we like to call her Irene instead of Irene. But she's Spanish, but her English is amazing. How long did you spend in the U.S.?
I spent 5 years living in the United States, and I was 8 years old when I moved and 13 when I came back. So
where did you go? Where did you live?
I used to live in Potomac, Maryland.
Potomac, Maryland, yeah, the DMV. That's where I'm from. Cool. Okay, so what is your background, scientific background?
Well, I'm a biochemist.
Okay.
I have a degree in biochemistry, and right now I am doing a master's degree in biomedical research at the University of Navarra in Pamplona.
With a specialty in
neuroscience.
Neuroscience, yes, that's why we're here. Cool. So today we're going to talk a little bit about Parkinson's disease or Parkinson disease. They cut off the S recently. It's been bothering me. But Irene did a project on Parkinson recently for your undergraduate.
It was a diploma on research training. So I spent 2 years in a laboratory that are doing currently, and they did it back then and still doing it, research on Parkinson's disease. And so I took on a project that they were working on and worked very closely with their researchers. So cool. I'm here to talk about that today.
Who are the researchers? Maybe I know some of them.
Well, basically there are two doctors from the Clínica Universidad de Navarra. Mm-hmm. So one of them you may know, Mar. And then there's another.
You worked with Mar? I didn't know that.
There's a fellow biochemist in the lab. His name is Ivan.
Cool.
Okay.
So tell me, Irene, what is Parkinson?
Okay, so I know that in Straight from a Scientist, you have had various podcasts about Alzheimer's disease.
Yeah.
But I am here to talk about Parkinson's because even though it is not as common, it is still the second most common neurodegenerative disorder. And it is a disorder that is basically characterized by bradykinesia, which means slow movements, and rigidity. So patients are very rigid when they move their limbs. Also tremor, which is a very common characteristic. Most people are aware of this. And also postural instability. So it is very common for them to fall. And so this is like the main 4 symptoms of the disease. But recently, over the years, that has been developed that a theory that says that Parkinsonian patients don't only have these motor symptoms, but they also have non-motor symptoms that affect various systems, such as the digestive tract, or very common is the loss of olfactory, so their smell, their smell is reduced, their capacity to smell.
Now, I remember learning in one class that if you start to lose your smell, it's kind of like a sign that you might have Parkinson's in the future.
So what it's been seen is that these non-motor symptoms actually may appear
beforehand.
A dozen years earlier than the motor symptoms. Okay. So they could be a predictive factor, except that they're not very specific. Yeah. So losing your sense of smell may come from very different things,
but it doesn't mean for sure you're gonna have Parkinson's.
No, it doesn't. No. Don't worry.
Yeah, another one I want to bring up was the REM sleep disorder. That's a big one that is pretty predictive. Not predictive, but it's a risk factor for Parkinson's later on.
This may also be one of the motor symptoms of the disease, and that also does come earlier in time than the diagnosis of the disease. So it's predictive, but it doesn't mean that a person with REM sleep disorder will automatically have Parkinson's disease. So they have to establish a correlation between these patients that may be at risk and the ones that will actually develop the disease. So this is one of the important things that research scientists are working on right now, is developing tools in order to detect which patients are the ones that will develop the disease.
Now, are there any genetic factors to Parkinson, or is it strictly
no?
Or we don't know.
So normally 90% of the cases are idiopathic, which is just a fancy way of saying that we have no idea why they're caused. So yes. So when you hear idiopathic, don't think anything fancy, because it just means that.
It just means we don't know.
Yeah. So these patients, these idiopathic patients, do seem to have a genetic component, but it's not as important as the 10% of the cases that are familiar forms, which means that if your parents or your sisters or your siblings, whoever has the disease, may pass it on. So normally these familial forms of the disease actually have an earlier onset. Normally Parkinsonian patients develop the disease around 65 years old, and however cases of genetic importance with familiar forms, they develop it earlier normally. So this is another clue as to what form of the disease it is.
So, okay, the familiar genetic forms would be they have an earlier onset.
Earlier onset and a very clear genetic mutation. So, for example, you may know the actor Michael J. Fox from Back to the Future.
Yeah, of course.
Yeah, he has a familiar form.
I didn't know.
He developed that very early on, and his case is actually familiar.
Interesting. Now, other than REM sleep disorder and losing your sense of smell, are there any other risk factors that can promote the development of Parkinson's?
Well, of course. For example, here is a very interesting story. In 1970s, in the San Francisco Bay Area, they had a lot of cases in which 20-something-year-olds, very young people, were coming in with Parkinsonian symptoms. However, they didn't have any mutations, and there was no reason for them to be having these symptoms, and it was actually because they were heroin users. And what they had was they were using homemade drugs, and one of the subproducts was a toxin that attacked specifically the neurons that die in Parkinson's disease. So they had the symptoms even though they didn't have the genetic or the normal course of the.
Now, do we know if they got Parkinson's disease later on in their life?
Well,
or was it just the symptomology?
It was a mirror.
It was a mirror image.
Yes, because it actually produced the same neuronal death. So the same regions of the brain and the neurons died, and once neurons have died, well, they can't be recovered.
They can't come back.
So
slowly.
Yes, they were treated as Parkinsonian patients. Yes.
Now, this is actually can be positive for us because now we're using certain models. You were telling me before the show that we're starting to use this toxin as a model for study.
Thanks to this sad event. Yeah. Yes, one of the best animal models was developed, which is the MPTP model. MPTP is a toxin that causes the selective death of neurons in the substantia nigra. This is the region of the brain that is affected in Parkinson's disease. So what they do in the animal models is they inject them with the disease, and specifically in primate models, which is what I've worked with, since they have such an anatomical and also behavioral similarities with us humans, what we see is that when the neurons die in the substantia nigra, they show the same symptoms. Even though they don't have the same molecular pathology, the symptoms are reproduced. So it is very useful to study symptoms and see how they react to different treatments.
So this is good for developing drugs to treat the symptoms, but we're not actually treating the cause or the root cause, which we still don't even know.
No.
Right?
So right now, the only treatments available for Parkinson's are aimed to relieve the symptoms. There is no cure for it.
We don't know enough yet. So getting into that, what are the current treatments and what is their efficacy?
Well, as I said, they don't cure the disease, they only treat it. So the main course of action for this disease is Ldopa. Ldopa is a drug that is supposed to raise the levels of dopamine, which is the neurotransmitter that is missing in Parkinson's disease. So right now, what they do is they inject them, or they take them, the patients, and this raises the blood levels of Ldopa that is crosses the blood-brain barrier, and in the brain is transformed to dopamine. So it just replaces the levels that have been diminished due to the neuronal loss, but it doesn't treat.
Yeah, exactly.
It doesn't cure it. The neurons are still going to die.
Yeah, now, so Ldopa basically is a precursor to dopamine, right? It's basically the building blocks for dopamine in the brain. And like Irene said, is that it solves or it fixes the symptomology of Parkinson's. But the problem is, like most drugs, you start to build up a tolerance, and the effects start to.
Well, it's not just a tolerance, is that because it doesn't stop the neuronal loss. Patients, even though they do have, at first they have this honeymoon phase in which, because they still have around 30% of the neurons left, these levels of dopamine relieve the symptoms. However, as the disease progresses and the neurons are lost with a higher rate, in the end, there is just simply not enough neurons to store the dopamine and to regulate its levels. So in the end, these treatments actually have secondary effects such as dyskinesias, which are involuntary movements that are the main cause for patients, for secondary effects in patients. They're very serious, and they do require also treatment. So in the end, it's not very effective, and what they're seeing is that there needs to be alternative therapies.
Yeah. I remember in one of our classes, we were shown a graph of like the dopamine levels and when the patient was taking the medication, and it was showed like a huge spike and then a huge drop, and it just, compared to the normal levels, you kind of get like, you either get too much dopamine and then it starts to drop lower than what you need, and it's basically, it's not a perfect cure, but I have to be honest, it works well. I remember I have one of my friend's godfathers has Parkinson. He was the first person I met with Parkinson, and I remember talking to him, and we were chatting, and you could clearly tell he has Parkinson's. He has the tremor, the tremor at rest. And I remember him taking the medicine, and almost instantly the tremor disappeared.
Yes.
It was like his movement was normal.
That's what they call the on phase.
The on phase, exactly.
Right, when the patient seems almost asymptomatic.
Almost normal.
Yes, like he would not have a disease.
But the sad part is that this drug eventually loses efficacy, and after months and years, you start to, they don't get the same fix as they did before. So basically right now, I guess we're looking for new treatments, and we were talking about this a little bit before as well. Let's talk a little bit about some of the new treatments, because Ldopa is good, and at least for a couple of years, but it's not the end all be all, and we really need to find something new.
Right. So there are other pharmacological approaches, such as a dopaminergic, drugs that bind to dopaminergic receptors in order to substitute dopamine.
Agonists, right?
Right. Or other drugs that will help to raise the levels of dopamine that actually cross the blood-brain barrier. But these are all dependent on Ldopa, and again, their efficiency is really reduced. So what they're looking at now is a surgical approach to Parkinson's, and this is called deep brain stimulation. So basically what it does is that they inject electrodes into the subthalamic nucleus.
So they have to do a surgery, is what you're saying?
This is a surgical procedure, yes.
Well, one quick thing I was going to say about the drugs is that these drugs they're more a global approach. So when you take a dopamine drug, you're not just getting dopamine in the place where you're missing it. You're getting dopamine throughout the whole brain. So,
dopamine regulates various aspects of the body, and it's not pinpointed exactly at the substantia nigra. It can go everywhere in the body.
So this is one of the problems with the pharmacological approach, is that you're getting dopamine everywhere in the brain instead of just in one point where it's missing. So the cool new thing about deep brain stimulation, which, well, you can tell me about that.
Well, it targets specifically the subthalamic nucleus, as I said. So this is one of the nucleus that is affected in the circuitry involved in the control of movement. So what they do is that these electrodes are this equipment, this surgical equipment that regulates electrically that nucleus, because the brain, as most of you already know, works through electric connections. So what it does is through it modulates the electric connections within the subthalamic nucleus in order to inhibit it, and that way it will reduce the activation of this pathway that is responsible for movement. That way patients will have less rigidity, less muscle contraction, less tremor, because the muscles will be more relaxed. There will be less movement, and it will allow for normal movement to be it will allow for the normal movement that the patient wants to do, and it will inhibit that tremor and that rigidity that the patient has as the basic symptoms.
Let's explain this a little bit better, because you're saying that in order to do this, they have to do a surgery into the brain. So they're implanting a chip or something, some sort of
It's
electrode.
an electrode.
It's
an
electrode. And they put it right on top of the subthalamic nucleus.
Right. So that is a deep nucleus within the brain. Yeah. So they have
the problem is it's almost in the middle of your brain. So in order to get there, they kind of have to...
They have to do a neurosurgery cross.
And you've seen one of these, no? You were...
Well, yes.
You were participating
I have been in surgery, seen one of these. So what they do is they inject them through the back of your head, and they open up a little space in the cranium, in the bone.
A small hole?
Yes, a burr hole. And what they do is they inject the electrode. So this is actually a two-step surgery, because first they do this whole implantation of the electrode, and then once it has been implanted, they do a second surgery, because of course these electrodes, they require a battery. They're electrical apparatus such as your phone, so they do require a battery. So what they do is that they put the cables that connect the electrodes, they pass them behind your ears under the skin, and the battery is placed under your armpit. So
inside of your armpit?
Yes. Inside, under the skin, so that way you have them completely implanted inside of you, and you can go home and the symptoms will hopefully go away.
Now, have you heard, what are the success rates on this surgery?
Well, I've heard that they are having good results, that patients do see the difference. They don't have these on.
Now, it's a constant stimulation of the subthalamic nucleus.
Yes, you remove this on-off phenomenon that you have with the drugs.
You have a more baseline.
It is always constant.
It's a constant rate. That's nice. Okay.
So what they're seeing now is that maybe it's not as good as it sounds, because you do need activity in the subthalamic nucleus at some point.
Exactly.
So in the end, you can inhibit or activate. You don't really know what you're doing when you're sending in these electric waves and this electric modulation. So what they're trying to do is perfect the electrodes that they implant in order to make them more controllable, in the sense that you can modulate the activity that you want within the nucleus in the different parts even.
Well, they need to look at, a normal subthalamic nucleus, find out the baseline. And it would be cool to see that if you had like an application on your phone and you could see your levels and possibly say, okay, I need a little bit of stimulation, I need some inhibit, you know what I mean?
That would be great. I think patients would thank you a lot, Julian, if you did that.
But the problem is these old patients and they don't know how to use their phones.
Not all of them.
The next generation.
Yeah.
But that's interesting. Okay. So that's what they're working on nowadays.
That's like the latest.
That's the latest hot topic. Yeah, I've heard that from some other people. Are there any other things that they've been working on, or?
Well, I wanted to add something, that this deep brain stimulation has been done in other nucleuses to treat other diseases, such as, for example, schizophrenia. They're doing it too. So it's not only...
Where do they stimulate in schizophrenia? I don't remember.
I don't remember.
That's okay.
But I do know that they're doing it, and that they're having also good results. So what they're doing, they're applying it for different nucleuses, because in the end, they've seen that there are circuits implied for each disease. So what they're trying to do is modulate them electrically.
Yeah, and this is crazy. Speaking about electric modulation, I have heard that there's a company that's in the US that's selling kind of like a transcranial stimulation. We were talking about this the other day. Yes, I've seen it. And it's supposed to stimulate your premotor cortex, which is the part of your brain that anticipates movements. And so their thought is that if you stimulate it before doing an activity, a motor task, that you're going to learn the motor task or do it in a more effective manner. So a lot of athletes are buying these. They kind of look like headphones, and these kind of, I'm not sure.
It's very interesting.
It's very interesting. Yeah, I don't know a lot about the science behind it, but I just thought that was interesting, is that you're sending electrical signals to parts of your brain in order to like activate them at will instead of...
In the end, it's crazy how something as important as your brain and its brain activity, how it can actually be modulated by an apparatus.
Exactly. I think this is the future. I think, it's starting to come out. Also in depression, I remember in Manolo was telling us about they have the electrical stimulation for depression, severe depression patients, not your average sad person.
No, I'm not sure that would work.
Didn't get sobresaliente in the test. But interesting.
Yes.
So what do you think about the future about just stimulating the brain? But what am I trying to ask you? What do you see in the future of this electrical brain stimulation?
I think that in the future they will develop, we need engineers in this field, and we need them
Sebastian.
We need them to develop good electrodes and better ways to do a fine regulation of the activity and the modulation that we want, because in the end this is a very big approach in the sense that you are doing this surgery in which you're implanting an electrode in the brain, and you just leave it there. So it would be great if we could modulate the activity and how much we want the nucleus to be activated in that sense. So what I see is that this has a lot of future, but it still needs a lot of technical and also research work. We need to know what the patterns are and how are they affected in Parkinson's. So there's still a lot of room to grow.
Yeah, not even just with this disease. I think this is a whole field that's going to open up, like neuroelectronics or something like this. That's crazy.
Yes.
Yeah, I think it's kind of scary too. We don't really know that much about each of these. We know the subthalamic nucleus does, plays a role in the basal ganglia, but we don't know exactly what its complete role is.
Exactly.
We know the thalamus has like 100 roles, but so it's kind of dangerous, and moving 1 centimeter or 1 millimeter to the left or the right, depending where you electrocute or stimulate it, could make a huge difference. So.
Also, they do say that it's organized topographically, which means that different regions of the nucleus will control different regions of the body. So maybe you're hyperactivating the arms and not the legs, and yes, so that's crazy. We do need more.
And it's not the same with everybody. Is the other thing. It depends. The brain is plastic, like we all know. It depends. Where you grew up, what kind of activities you did. If you played golf, if you went horseback riding. This is crazy. So.
Yes. So it's the future, but it needs work.
It's the future, but we have a lot more to study, and it needs to be always
it's a project,
an individual approach, because the brain is quite individual. Cool. Well, what else do you have to tell me?
Well,
Irene?
I was going to talk about what I did with my project.
Tell me about your project a little bit. We talked about Parkinson's. We talked about where we're at right now. And tell me about your project. What kind of stuff? What kind of research did you do?
So I'm going to start with very basic concepts of Parkinson's. Is that same as other neurodegenerative diseases, it is characterized by an aggregation of proteins. And the protein that aggregates is alpha-synuclein. So what it's been seen is that alpha-synuclein has these post-transcriptional modifications, such as phosphorylation. So
what is phosphorylation for someone that doesn't know?
This means that a phosphate group is attached to the protein. So there are other proteins that come in and do this work, and they are called kinases.
Okay.
Right. So what it has been seen is that alpha-synuclein that is aggregated has more phosphorylation than non-aggregated alpha-synuclein. And we don't really know what role this has in Parkinson's disease.
Mm-hmm.
So what we wanted to do in our approach was find out everything we could about this phosphorylation and the role of the kinases involved in the phosphorylation. So one of the theories that exists right now in the neuroscience field is that aggregation is due to a disruption in the clearance mechanisms. So this means that the cell has different pathways to get rid of proteins that it doesn't want anymore, or even organs. So what seems to be happening in these neurons in Parkinson's disease is that these mechanisms are affected. So the guy who takes out the trash doesn't want to take out the trash, in general terms. So what would happen is
the paro. He's on unemployment.
Right. So what would happen is that since the proteins are not being cleared, they aggregate, and maybe this is not known, but maybe this would lead to neuronal death. So what we wanted to see was this kinase that is called PLK2. And PLK2 has been seen to both be involved in the phosphorylation of alpha-synuclein. So it comes in and it attaches these phosphate groups to the protein. But also what it does is it acts as a chaperone. What it takes the protein, it binds to it, and it leads it by the hand to the clearance mechanism. So this was proposed by a research group, and we wanted to see if this was happening in the MPTP monkey brain. Because as I said before, MPTP causes death of neurons in the substantia nigra, but they don't have Lewy bodies. They don't have this alpha-synuclein aggregation. So we suppose that there must be something in the monkey brain that is not allowing for alpha-synuclein to be aggregated, while in the human brain it exists and it isn't replicated by the toxin. So what is it? That was our question. So based on the studies that they had done with the PLK2 and autophagy, what we did is we studied the levels of both alpha-synuclein and PLK2 in the substantia nigra and other regions of the brain. And in the end, what our results were is that alpha-synuclein levels of expression, so the protein was not producing more alpha-synuclein, but it was producing more PLK2, more kinase. And our theory is that since alpha-synuclein is being aggregated, there are signals that stop the production of more alpha-synuclein. The neurons say, stop, we have enough. While there is another mechanism that says, produce more kinase, we need more kinase. So you have more production of the protein, but then when we studied the protein levels, they were actually reduced. So there were less levels of PLK2. So our theory is that alpha-synuclein binds PLK2, it phosphorylates it, and it leads it to the clearance mechanism in order for the neuron to get rid of it. And this works perfectly well in MPTP monkeys. They don't have Lewy bodies. However, our theory is that in Parkinsonian patients, they have a problem with this. So what we propose as a future treatment target, and also more research needs to be done, but we propose that maybe we should study what the autophagy system is like in these Parkinsonian patients, because maybe what they have is a problem with how proteins that are maybe phosphorylated, so they don't work well and they are aggregated, how these are removed from the cytoplasm, from the neuron body, and maybe they have a problem with this. So we think that it's a very interesting approach, and it was very interesting what we found. And we want to see if this is happening in humans too. This would be a great approach.
So there's further studies need to be done.
Yes, a lot. But it's a good field. It's a good approach. And I do have faith that maybe if we keep working in this line, we can see more alterations and help build the bigger picture of what is going on. Because in the end, it's such a common disease, and we don't really know why these neurons are dying, and why are they dying in the substantia nigra and not in other regions? Why are they selectively dying here? So we think it would be a great idea just to get the whole picture before.
Yeah, I think the first step is we need to know what's going on,
Mm-hmm.
and then we can decide, okay, how can we fix it? But if we don't know what's going on, the only thing we can fix are the symptoms. Is
Exactly
what we're doing now.
That's what we're doing
And
now.
as we can see now, that only gives you a couple of years, 3,
Mm-hmm.
4, 5 years maximum of symptom relief.
So
Interesting.
a lot of research is also being used in order to do an early detection of this. I saw today a video actually of a man that had developed an application for early detection. So what they do is that they've seen, just as your hands may tremor or you may have rigidity as a Parkinsonian patient, this also happens in your larynx and your voice. Yes. So when you talk, it is also affected. So what they do is that you would call in this service and have like a 30-second questionnaire in which they ask you to talk, and from what the noises you make and how your whole system of talking and the muscles used, how they work, they can detect this.
Are you serious?
Yes.
Wow, that's
early
crazy.
detection. Yes, but I think that this is a great approach, great idea. However, why would you want to know earlier in time if you can't cure it?
Exactly.
So right now, I think that most of our efforts have to be on why is it caused and how can we find a cure? How can we stop it? Maybe not revert it, but maybe stop it would be a good idea. And that's when
prolong the onset is huge. At least in Alzheimer's, we know, okay, if you do intellectual activity and learn languages, has been shown to delay the onset of Alzheimer's. Well, we don't know anything about that in Parkinson's.
No.
We have no idea. Interesting. That's really interesting. So what about you? What do you think you're going to do next after the master's?
Right. Well,
are you going to continue with Parkinson, or?
I do like this field of Parkinson's, but right now I have moved on to neurodevelopment
issues.
Yes. So what I'm going to be doing is specifically autism and schizophrenia. So what we're going to be doing is studying also, as we said, it's important in Parkinson's, in these diseases, the brain activity and the waves, and it's very important. So I've actually become very interested in the electrophysiology and the circuitry involved in diseases. So right now, that's going to be my plan for the future. Although I am passionate about the field of Parkinson's, I can't lie.
Yeah.
I have both professional and personal reasons, and I do like this research field. So I will be moving back and forth, I guess.
We'll have to have you on after your project
I'd be happy to come back.
To Switzerland, right?
Yes.
That's awesome.
I'm going to live in Basel.
And you leave in about a month or a couple weeks?
A month.
Cool.
Very
excited?
Cool. Very excited. I think I'm going to go on to a great team. They seem to be doing...
They seem to know a lot.
Yes. They're going to be great, doing great stuff, great things. So I'm just happy to be a part of it and learn everything I can while I'm there.
Awesome. And PhD, you're thinking about where? Staying here?
Oh, don't ask me that
don't
yet.
Ask me this yet? Okay.
A year may change a lot of things.
Okay. I won't ask you yet. Well, we'll have to have you on definitely another time after your research project this summer, and to hear your results and what you learned. Cool.
I'd be happy to do it.
Well, anything else to tell me or tell us? No? Okay. Well, thanks for coming on today, Irene. It's been a pleasure.
Thank you for having me.
Yep. And if you want to contact Irene, you have any sort of ways we can contact you, or listeners, if anyone has questions or wants to
we can post it on Facebook.
We can post it on Facebook.
Yeah, my contact info.
We will put her contact info in the show notes so you can follow her on Instagram or do whatever. Okay. Cool.
Okay, great. Thank you.
Thanks for coming on. Bye-bye.
Thank you for listening.